Good morning. My name is Michael Edwards. I am CEO at Bioinfo Solutions. This is my own company where I do consulting for bioinformatics. I used to be an assistant professor here about 4 years ago. I got really sick of writing grants that didn't get funded and all-day faculty meetings, and decided it was way more fun to do science rather than all the administrative crap. I guess what I'm basically saying is, it's all about numbers. And to analyze numbers, bioinformatics kind of, is associated with numbers coming from biological systems, but, we get numbers everywhere. And so the skills you learn in bioinformatics, you can absolutely and utterly apply to just about anything you want. I've actually used bioinformatics to try to figure out NFL games. I did pretty good, actually. I still didn't win the pool, dang it. But, okay. So what are we going to do today? So first off, I think everybody has an idea of what bioinformatics is, but probably doesn't really know what it is. So we're going to talk about, what is bioinformatics? What is some of the data that we use to, make these associations? And then we're going to, after my presentation, we're going to have an interactive demonstration. So you all are going to follow along with me, and we're actually going to do a bioinformatics exercise. And as we go, what I want you to do, instead of taking notes, is bookmark all these places that we go. And so that when you get to your own, research, you can go back and actually find these places and use it for your own research. After that, once I've kind of demonstrated how we do this, I'm going to let you explore on your own. And basically, I'll be here in the room to take questions and help people along the way. I'll also present maybe some other things that I think are important in bioinformatics. And then, if people have results from their own independent exploration, maybe we can discuss these results. So what is bioinformatics? So this is a part where we, class participation. What do you think bioinformatics is? Anybody? What's your idea of bioinformatics? Somebody.
Numbers.
Just numbers?
You just said it's all numbers. So.
It is numbers. Yeah, it is true. What else? What would you do in bioinformatics? What would you use bioinformatics for in your research?
Making graphs and Excel.
Yeah, or is that biostatistics?
Oh, I don't know.
Right. What about you? What would you do what do you think you would use bioinformatics for in your research?
I've always thought of it as you might use it to analyze genomes and sequences and similarities and things like that.
Yeah, absolutely. That's absolutely a part of it. What about you?
Maybe you could create models, computer models of a biological system.
Yes, absolutely. I would say that's more computational biology, but absolutely bioinformatics involves that in making models. By the way, there was this famous statistician that said, all models are wrong, but some are useful. And that is absolutely true. You can never, ever make a model that's perfect, right? You just make something that hopefully can benefit you, but realize at the heart of it, it's not going to be right. What about you? What do you think bioinformatics is?
I would say probably data analysis comes similar to taking a broad set of data that doesn't really have too much specification, trying to find some information from it and use that in further research.
Yeah, no, absolutely. Well, what about you in the back? Last one.
Everything.
Everything I said. What about blast searches, right? Similarities, sequence data, right? How do you turn that sequence data, those reads that we take from all these genome sequences, and turn that into numbers, right? That's what I do. That's a big part of what I do. So again, maybe taking biology and turning it into numbers and then using those numbers to analyze something. Well, here's my kind of simplified version of what I think bioinformatics is. It's kind of represented by these pictures. And this came to me, when my daughters were pretty small. Actually, my oldest is graduated high school this year. But we used to go to Denny's, and you remember they give you those placemats, to keep the kids all calm. So, they Parents could eat, they have a little, well, play your game, there's games on there. And they always had that one game, they'd have two pictures and they'd say, okay, what's different between these two pictures, right? In a way, that's exactly what bioinformatics is, right? I have two things that look pretty much exactly alike, right? What is the actual difference between there? What pattern is in this thing that isn't in this one? And at its very simplest form, that's exactly what I'm doing in bioinformatics. And I can see you all kind of looking at that picture, a bunch of you trying to figure it out, right? I mean, this is a human, function. Like, this is what we do. We are natural pattern recognition, creatures, right?
They all want that placemat in front of them.
Yeah, can we just get the pictures and not the lecture? But it is, honestly, I mean, that's kind of how I see it, when you look at, when we look at the genetics between individuals, we're like 99.6% identical, right? Most of the stuff between us is the same, but obviously there's some differences there that cause huge, can cause disease, can cause huge phenotypes later on in life, right? So how do we recognize that? And so at my simplest explanation, that's exactly what I'm doing. You can almost see all this information as like pixels, right? If you look at your average high definition TV, how many boxes do you think is in it? What do you think? Make a guess. No? 8 million. Actually, my TV doesn't have 8 million because I don't have a very good TV. But most 4K has 8 million boxes, right? So when you're watching your TV screen, do you see all those boxes? No, of course you don't, right? And if you do, maybe you should probably work for the government because that's not normal, right? It's our mind puts all those pixels together and it forms a picture, right? And these pictures can make you laugh and they can make you cry, but in actuality, all you're watching are boxes, right? And if you really want to get to the simplest part of that, all you're watching really is zeros and ones, right? So my idea is, how do we take all this information, these pixels of information, and basically create a picture? And that's actually what I do in bioinformatics. That's what I feel I do. I take all this information and I put together graphs, I put together, heat maps, all these, pictures of the data that I can give to my collaborator and say, hey, here's your disease. I think you should focus on this, right? So that's kind of how I see bioinformatics is and my role in that. So where do all these pixels come from? And there are a ton of pixels. It's a waterfall and it just keeps coming and coming. It's like, it's just, we are generating exponential amounts of data every day, right? And for my mind, I love this. Like, we could stop sacrificing mice, growing cells, running Westerns, and I'd have enough data for 3 careers easily, just with what's out there, right? And that's one of the great things about bioinformatics is I don't have to rely on the data that I generate or my clients generate. I can go out on the public web and get whatever I want. So where's this data coming from? So when I was in grad school, we used to basically bow to this, the central dogma of biology. I'm sure everybody's heard of this. You have DNA, transcription, translation, protein, right? This is always a dogma. So of biology. So, but if you go in here, I mean, if you look at this, just starting with the DNA, right? And in humans, we got 6.4 billion nucleotides, right? In this DNA are in humans, about 25,000 different genes, right? So, and they will produce messenger RNA, which is translated into proteins. And just in its natural state, there's about 500,000 different proteins. Just this data alone is enough to overwhelm your average scientist, right? They see this and they're like, there's no way I know what to do with all of this. But there's more, right? We also have microRNAs, right? Everybody knows what microRNAs are. Oh, it's nice presenting in front of college students. I do this sometimes to the general public and high school students, and when I say microRNAs, you're just like, what? Are you kidding me? So, there's these little bits of RNA that bind to the messenger RNA and can cause its destruction. It's a level of modulation of our expression. And again, we can measure all of these different microRNAs, right? And we go to the messenger RNA. We have 25,000 genes. Does that mean we have 25,000 versions of messenger RNA? Yes? No. No, of course not. Right? You have splicing, you have introns, you have exons, right? You can shuffle these all around. At one point, I was working on a gene that had 11 different versions of itself. And they all did something different. It was an absolute and utter nightmare. I'm sure you've been there, John. It's just terrible. Like, which version is it? Is it that isoform? Oh, man. So not only, can we look at the 25,000 genes, but a lot of those have its own isoforms, and we can also measure that as well. Go back to the proteins. Now, proteins are a mess. I did a postdoc in proteomics, and I got the heck out. Like, it's just, anybody here working on proteomics? Doing proteomics stuff? Okay. So I can bad talk them is what you're saying. Okay, great. No, it's not, I'm not bad talking them at all. I mean, it's something, people should absolutely study, but it's tough. That you could take a protein, you put a phosphorylate it, put a acetyl group, you can modulate this in many different ways and get all kinds of different proteins. So again, it's very difficult. But we can measure all this. Let's go back to the DNA, right? You can do all kinds of things. We just talked about epigenetic changes to the DNA, right? You can, on the histones, you can methylate them, which causes it to condense, right? Again, we can measure all of this secondary structural changes to the DNA. And as I said, these structural changes are important. And in fact, I think it's probably the most important part of the regulation of genes in your genome. As an example of this, can everybody see that? There are two bugs up there. All right, the one on the left, or the one on the right, is your basic grasshopper. Hops around, stays pretty local, pretty mellow. You might be riding your bike on a trail and jumps in front of your face, right? It's just a grasshopper. The one on the left is locust. Now these things, shorter, different color, orange, more aggressive, tend to form swarms and can travel thousands of miles and devastate entire communities by eating all their crops. These two organisms are genetically identical. The only difference between those is the way their chromosomes are compacted, right? Again, all of this stuff that you're looking at, we can measure. So as you can imagine, when all this stuff started coming about, your average scientist who isn't trained in data science just kind of freaked out, like, oh my gosh, what am I doing? Go ahead, have a seat. Martin? Okay. So again, all of this stuff we can measure. This all contributes to the pixels that we can choose from to build our picture. So again, what do we do with that? Your basic experiment, in order to exploit this, right, is, you take, say, a bunch of people that don't have this disease, that are phenotypically normal, and then you compare them to, say, a group of people, say, with a particular disease, right? You maybe measure all the gene expression that's going on in those individuals, right? Use, say, mathematics to determine the differences between these two, like what genes are different, and this can be a variety of statistical tests. And then usually you get a list of genes that are different, right? This, for me, is the fun part. It's very easy to get to this part, actually. It's, the actual statistical tests that we use to look at the differences, usually aren't that sophisticated. It's what you do with this list of genes that's important. And it's not only just, we're not just looking at the differences between, say, a control versus disease, but we're also looking for similarities as well because it tells us a lot of that. So given up here are examples of genes, gene expression. Each one of these rows or these kind of blocks here represent a particular gene over many different samples. The height of the bar would signify its relative activity. So the higher the activity, the higher the bar. The lower the activity of that gene, the lower the bar. So if we're looking at these two, so these are two separate genes here. What do you notice about these two genes? Like when it's high here, what happens here, you know, and going, you know, looking at when it drops here, what happens here? Can you see that they're kind of related, correlated? When you spend enough time looking at like patterns with me, like it's pretty easy to see, but I think for some people, like, plus it's pretty small up there. But if you look here, like at the very end, you'll see this, you know, it's lower here, lower here, and then much higher here. Lower, higher, lower, and then much higher, right? It's kind of that similar pattern. So what would you say about that? When you have all these different conditions, say, or samples, and you see that one gene expression, you know, when it tends to be high in that particular condition, this gene is also high. When it's low in another condition, it's low. What would you say is going on between those two genes? What do you think? Any ideas? What do you think they're doing? Any ideas? None. Working together, maybe? Right? Maybe they're super friends, right? I can always tell how old my audience is by this picture. I'm surprised. This is what I used to watch on Saturday mornings, right? This is back when Robin didn't have pants. Kind of makes you wonder about Batman. Okay. So, I mean, basically, you know, what you're looking for is, you know, this correlation, you know, they're probably working together or doing something, right? It's like if you saw somebody, if you saw two, you know, you knew two people and they're always together, and if you saw one, you always saw the other, but if you didn't see one, you didn't see the other, would you assume they're friends? Yeah, right? Why would they always be hanging out with each other? Same thing happens with genes. If we correlate, if we find things that are related, they're probably related in a similar function. And they're all probably working together. I'm going to tell you right now, like no gene works by itself. I hate the term driver mutations. I just hate it. Because it signifies that one mutation is causing all these things. And it's absolutely and utterly crap. It absolutely doesn't, right? Everything works together in networks and gene expression, you know, pathways. And if you don't understand those pathways, you're really never going to understand your system. So that's why finding things that are similar, you know, is good. And here's the other aspect, right? So here you can see in this gene it's really high, and in this one it's really low. And then when it gets high here, it seems to get low here. What do you think's going on there? What do you think?
Maybe have like opposite functions.
Right, maybe one is suppressing the other, right? In order for it to do its thing, you know, once this goes up, then this one gets suppressed, and vice versa, right? Maybe they're, you know, Legion of Doom, maybe they're enemies, right? And that happens a lot, and we're going to see that in cancer, and I'm going to talk about that in cancer, is that it's not just oncogenes stimulating growth, but you also have another force that's trying to suppress that, right? Your tumor suppressors, your apoptotic, your DNA repair, trying to stop that, right? It's a battle of two forces. So I think the easiest way over the years I've found to try to explain this is to kind of, you know, to explain bioinformatics is to set it up as an investigation, like a police investigation. So follow along in my example. You know, basically what we're going to do is, I'm going to demonstrate, you know, what this kind of explain bioinformatics, how we approach it. But again, we're going to use an analogy to do that. So in this analogy, what we do is we have a bank right here in the middle. And all of these individual genes around here are people and all of them were walking by the bank or around the bank by themselves, didn't know anybody else, or said they didn't know anybody else around the bank, right? They were just walking by. The bank gets robbed. They hit the alarm, the police come in really quickly and basically lock down the whole area shortly after the bank's robbed, right? In this situation, where do you think most of the robbers are going to be?
Separated.
What's that?
Separated from each other.
Not necessarily, right? So the bank gets robbed. Shortly after the bank's robbed, they hit the alarm, everybody locks down, everybody kind of stays where they are shortly after. So where do you think most of the guilty people are going to be? Together, maybe around the bank, right? Oh, and you're the investigator. I know this is not a police officer. I used to use some other like old TV Private Eyes. Remember Columbo? Yeah. No one got that. I know. Everybody knows House though, right? Maybe? Okay, good. Trust me, there's going to be a lot more references you're not going to get. Okay. So like we were saying is, so the bank's robbed. Police come in, you would expect most of the robbers to still be kind of close to the bank because, it was shortly after. Although some of them say this bank robber, was a track star, and ran a lot faster. Or maybe this one had a moped close by and, got away a little farther. But most of them in general are probably going to be around the bank, right? So you are the investigator. And you have access to, police files, the FBI, all of this information. How are you going to determine who is guilty and who isn't? Oh, wait a minute. Sorry. So that's the thing is that so most of the people will be around the bank. And what we can do is we can build perimeters around that bank, right? And that's kind of what we do with statistics is like I'll draw a line and I'll say, anything with a p-value of less than 0.01, I'm going to take. I'm going to consider that those are the guilty people, right? But as you can see, as we draw this line, you're getting the majority of your individuals are guilty, which are in the red, but you can see all these white ones, right? These were just innocent people walking by. Right? You're still getting innocent people. These are called false positives. And this is definitely a bane, something you have to consider when you're doing these like mass analyses, right? When you're analyzing 25,000 different things, you are going to get false positives. And we're also, as you can see, as when we draw this perimeter, we're also missing some guilty individuals out here, right? We could also draw it even farther, right? Maybe we increase our p-value to, say, 0.1, right? We're getting more guilty people, but we're also getting a larger percentage of not guilty people, right? False positives. So a lot of that is when you're doing your p-value, you're trying to determine where do I put that p-value so that I can get the majority of guilty people and minimize the amount of false positives I have. Does everybody understand that? With the realization that the tighter I draw it, the more percentage of guilty people I get, but I'm also missing a lot of other guilty people as well. But here's what we do, and this is what most people do in bioinformatics, is they do false discovery rates. Has everybody heard of false discovery rates? Wow, FDR, my goodness. So p-values work on individual genes, and it's the probability you would arrive at that value by random chance, right? Like 10% of the time, I would just get this by random chance. False discovery rates are based on lists. So what a false discovery rate is if I grab a gene list here, so what they do is they draw their list very, very close, like say to the bank, right? They're drawing their perimeter very tight. And so to the point where they say, within this circle, I predict, or I've calculated that 90% of the people that I find are going to be guilty, with only 10% being a false positive. So that is what a false discovery rate is. And that's what people apply to this stuff all the time, is that when I'm doing so many tests, I generate error. And how do I minimize that error? Well, I draw my circle very, very close. Again, a lot of this can vary, and it's based on your own personal idea of what you want to see. So now we're going to talk about like the individuals associated with it, like the guilty individuals. So again, you're the investigator. The bank's been robbed. You know that there's probably a higher percentage of guilty people closer to the bank. What else would help you determine, not just on location, but what other things would help you determine whether these individuals might be guilty? Remember, you have access to the FBI, data files, all this stuff. What do you think? Yes, criminal record, right? Like, say you found somebody near the bank that said he didn't do it, or she. Women can be bank robbers, absolutely. But, you ask them, oh, I didn't do it. But then you can look at their criminal history and they've been guilty of robbing banks before. Does that make them possibly more guilty or less guilty? Right? More guilty. If you've robbed banks before and you just happen to be by a bank that was robbed, you might be guilty. And that's actually exactly what we do, right? We can look here and we can basically look up the criminal histories of these things, right? If a gene I know is associated with, I'm trying to see if a gene's associated with breast cancer and it's been associated, say, with lung cancer, the probability that's also associated with breast cancer also goes up. And that's what we kind of use, and this is what I use to determine, what gene should we look at, right? It's kind of like The Usual Suspects. Anybody watch The Usual Suspects? Nobody. Gosh, man. It's a great movie. You should watch it. Okay. And I do more than just watch movies. I swear. It's kind of like, a gene identified in more than one study. It's like, say, and obviously there's not just one bank being robbed. There's lots of banks being robbed, right? And I look and you say all these different banks represent a different kind of cancer, and that the same gene or the same individual just happened to be by 3 different banks as they were being robbed. Does the probability that it's involved in the bank robberies go up? Heck yes. Absolutely, right? You would not expect that by random chance, right? So another thing, that's exactly what we do, right? It's, okay, I found this gene. It seems to be involved in the cancer that I'm looking at, and it's also involved in all these other cancers. So therefore, my idea is that gene becomes way more important, right? When we're talking about cancer and the usual suspects, it's basically, it's the criminals versus the cops, right? So you have your criminals, which are the oncogenes. Do these oncogenes start off as criminals? No, right? Just like regular criminals. Like, you don't come out of the womb as a criminal. I don't think so. Right? You develop that, and that's kind of what cancers do. I mean, these genes all serve a purpose, and then somewhere along the line, somebody corrupted them, right? They turned them to the dark side, and now they're doing bad things, right? So what are some of these oncogenes? Give me some examples of some oncogenes. You guys are studying. It's okay. You can talk.
I think I know of one.
RAS. What are some other oncogenes? ERK, yes. Anybody else? Growth factors, right? We can just be growth factors, signaling molecules, cell cycle genes are huge. Things involved in angiogenesis. So it's not just about, cell division, right? It's supplying, those cells food to grow, right? So a lot of, cancers use a lot of angiogenesis, building a blood system to the tumors itself. Managing telomeres, right? If these cells, so a lot of times what you'll find is topoisomerase goes way up in a lot of tumors because you have to keep building on the ends of the chromosomes because you divide so much, you don't want to divide into the genes. So what they do is they actually, a lot of these tumors upregulate the, taking the ends of the chromosomes. And again, some of the big time players, KRAS, EGFR, MYC, VEGF, some of the big players. And again, it's not just the oncogenes. If you've got criminals, you got to have people chasing them or trying to prevent what they're doing, right? So we've got tumor suppressors. What do you think the tumor suppressors do? Like, what's their average function? What do you think?
Trigger apoptosis.
Yep, apoptosis. What else?
DNA damage repair.
DNA damage repair, which goes into apoptosis. What else? Cell cycle checkpoints, stuff like that. Inhibitors, DNA repair enzymes, perfect, telomere maintenance, and again, programmed cell death. And if we look at some of the usual suspects in cancer, I would say the biggest cop in your system is P53 for sure, right? But you also have BRCA1, which is involved in breast cancer and DNA repair, retinoblastoma 1, and P10. And P10 is more the apoptotic cell cycle inhibitor. What I want to get the point across is it's not just oncogenes and it's not just tumor suppressors. It's the battle between these. And it's the forces of these two forces that when you're studying a cancer is what you really want to concentrate on. And if you just look at one side and not look at the other, you're missing it, right? So that's what bioinformatics, and that's what I'm trying to do in bioinformatics, is how do I tease out this relationship, right? This pull, right? When the oncogenes get, and that's the thing is, it's not only the police officers fighting the oncogenes, but the oncogenes fighting the police officers, right? They want to blind them. They want to take out some of their corpse, right? They don't want, it's this huge fight in your body. And, it's how can I study a cancer and then bring this stuff out so that once I know how the oncogenes and the tumor suppressors work, can I manipulate that system so that the cops win? Everybody get that? Okay. So what I'm going to do now is just kind of take you through, this is, I used to work on lung cancer quite a bit, so this is what the example you're going to get. Just kind of one of my examinations into lung cancer and kind of how we use this, these techniques to kind of figure the cancer out. So this was my criminal that I was after. I had a collaborator come to me and say, hey, we know this polo-like kinase 1 is really important in our system. We just don't know why, right? We see this upregulated in our cancers. We know it's probably an oncogene. What is it doing, Mike? Help us figure this out. Find its gangs, all that stuff. So polo-like kinase, it's a trigger for G2 to M transition, very much involved in cell cycle. It localizes to the centrosome, so it's in the nucleus. Also, if you lose it, you can actually induce apoptosis. So again, it's probably anti-apoptosis. And again, it's been shown, its criminal history, go into its criminal history, lots and lots of evidence of it acting in other cancers, okay? So again, you're the investigator. I was the investigator. I'm like, well, let's learn more. So with polo-like kinase, what you can do, this is a program I use. It's called Cohort Analyzer. It's commercial. I'm sorry, you can't use it. All the good stuff you have to pay for. I'm sorry. But here is what it's doing is, so this is kind of a percentage. This would be 100% of the samples in their database. But what this is showing is in all of these different samples, red would indicate an upregulated expression of that gene, and green would be downregulated. And these are all just different types of diseases. And you can see lung cancer at the bottom with 1,145 samples, and the vast majority of them showing increased expression of polo-like kinase. Look at all this. Kidney cancer. Some of these, you look at, I always see weird things with leukemia. So anybody here working on leukemia? No? Good. It's weird. It always has opposite effects, but again, you can see our criminal history. I go in, it's been convicted of lots and lots of crimes. And in fact, if we go to lung cancer, you can kind of see these as all different cities that this criminal is exploited, right? We go into lung cancer. What we can see is, close to over 95% of all samples in their lung cancer database have some kind of alteration to polo-like kinase. Either it's increased expression or there's a mutation in the gene, right? That tells me that based on its criminal history, this is absolutely and utterly a big player in the crime of lung cancer, right? So I've got my right, I'm pretty sure I've got my right suspect. So what crime has it committed? It's committed the worst crime possible, murder, right? If you look at, so these are Kaplanmeier curves. Has everybody seen the Kaplanmeier survival curves? Yes? I know, they're ubiquitous. I like them sometimes. I think you can manipulate them quite a bit. I've seen it, but. So here what we're showing is, so in this blue line, these are samples or individuals that had low polo-like kinase 1 expression, right? And they seem to survive a lot better than individuals that are expressing high levels of polo-like kinase 1, right? They tend to die a lot quicker, right? So again, the crime is there. I've got my suspect. I looked at his criminal history. I've seen what kind of crimes he's done. We can actually even go back even farther into his criminal history. And this is some work I did here on campus with the lung spore. But, you know, when you have cancer, there's a whole process before you actually form a tumor, right? And we've seen this on, you know, basically bronchioscopes. We can go in there and look at the tissue. This is what normal tissue looks like. When it starts getting this, then it starts getting this dysplasia. It starts kind of bending and moving, and the different levels like either thicken or narrow, right? And then finally at the very end, what it does is it can turn into a tumor. This is the only point where we actually treat the cancer. We've got all these other places. So what happens here is a lot of times you'll see this and you'll be like, uh-oh, it's going to be cancer, but then it'll revert back to normal. And that happens quite a bit. So how do we determine, you know, so can we use polo-like kinase to maybe determine whether we should maybe treat before it actually becomes cancer, or maybe just let it go because it's going to go back? Here is the expression as measured for the polo-like kinase 1 gene. In the red are the ones that actually go on to form cancer. Blue are the ones that get better. You can see a gradual increase as we go from normal to cancerous tissue, but you can see that in those samples with higher polo-like kinase 1 are way more likely to turn into cancer than those that have lower expressions of polo-like kinase. Did everybody see that? Right? This is part of bioinformatics. Can I not only observe what I'm seeing, but then can I use that expression to predict future behavior, right? And that's exactly what I'm doing here. So yeah, bad criminal. All right. So again, we're back at our regular, you know, our example here. Bank's been robbed. You know, the guilty people are probably close. You've also looked at criminal history, right, to see if they've been convicted of crimes. What other information would help you to predict whether a person is guilty or not? What do you think? Associations, maybe, right? What if these individuals were in a gang, right? So in this example, here are these individuals in the blue, right? Say they were just walking around by the bank, minding their own business, and it was robbed. They had nothing to do with it. But you find out there's a gang out there with about 7 individuals, and 5 of them just happened to be walking by the bank as it was robbed. Do you find that unusual? Heck yeah, right? Like, oh, just, you know, 3/4 of your gang just happened to be by the bank as it was robbed. Yeah, right. And you guys weren't with each other, you just happened to be walking by, right? It's these pathways, and that's what also helps us determine what's guilty and what's not. If you know of a criminal that's been guilty of a crime and he's got a gang, that gang is probably associated with that crime as well, right? Absolutely. You know, and these gangs can be huge, you know, they can be small and powerful, and they could be doing really terrible things in your body, right? Here's the thing with these gangs, though. Again, gangs, meaning pathways, is that they share gang members. So one person isn't just in one gang. You can have, an individual in hundreds of gangs. Like P53 is in lots of gangs or trying to fight lots of gangs, right? Or RAS or, some of these other ones, EGFR, right? So I always have, I one time had a lecture called Death to Pathways because I'm not a big fan of pathways. And that these gangs, just like the cops, we define these gangs, right? It's the police officers or, it's the police force that says this is a gang based on our intel. But what if the intel is wrong? And that happens a lot. Most of these pathways that you look at are 30 years old, probably developed by maybe 2 different groups using 2 different organisms, right? They aren't that accurate, right? They can help, kind of establish what's going on, but realize that the gang is way bigger than what the pathway is given. But again, it does help. It helps us determine, it's not only can we find the criminal, but then we find this gang, and what does that gang normally do, right? And that helps us define what's going on. I'm going to explain pathway analysis really quickly because I think it's important, right? And a lot of bioinformatics works on this principle. It's finding overrepresented biological units. So picture we're analyzing lung cancer, right? And we look at people that don't have the disease with people that do. We do the information, and we see what genes are different. And I have a feeling that this pathway is somehow playing a role in my system. This is how pathway analysis works in its very simplest form, right? If I find 152 genes, I do a FDR of 10%, and I have 152 genes that I believe are important in this aspect of lung cancer I'm looking at. And I want to know if this pathway is playing a role in this. The reason why all of this stuff works is that you have a limited number of genes to choose from, right? There's a finite number. So that, and what you can think of this is think of these 40 members as like pink balls in a sea of 25,000 green balls, right? And you get 152 draws from that 25,000, right? I get to draw 152 times, right? What do you think the odds of me just drawing one of these? Right? Probably not that unusual, correct? I mean, it can happen. But again, I'm drawing 152 times out of 25,000 genes. What happens if I get 3 now? Do you think that happens? Do you think that can happen by chance? Could, but it's probably unlikely. But here is usually what happens, is that I get 7 now. I'm drawing 152 genes. I get 7 genes from this pathway. The odds of that happening by random chance are infinitesimally small, which would tell me that this isn't a random act. That given the fact that I'm drawing all these genes, given the limited number of genes that I can choose or that I get, is telling me that this pathway is probably playing a role in my system. Does everybody understand that? You just know more about bioinformatics than just probably 80% of the scientists out there, I swear. This is it. And the thing is we can make these biological units anything, right? This is just a pathway. I can make this, say, genes in the nucleus, or I can make this genes known to be affected by a certain drug, right? I can put all of this information, I can survey all of that and figure out, are these things playing a role in my system? So we did that. So I did that, I had a bunch of lung expression data. These are genes that were correlated to histology, so they either increased or decreased as a tumor got worse, and these were the persistent versus regressive. So those lesions that went on to form lung cancer versus those that went back to normal. And what you find is, again, we're looking for those gangs, right? Here are all the gangs that I found in our gene lists. But look, the mitotic roles of polo-like kinase show up, right? That's PLK1's gang. I'm finding that, overrepresented in my gene list. Therefore, it's making me even more certain that polo-like kinase 1 is playing a huge role in my system. And here's its gang. So everything that is in red came up in my list. And you can see, it's just beautiful. Like, it's all cell cycle. You can almost see like where it's going. And the great thing about this, again, if I want to inhibit polo-like kinase 1, do I want to try to do it directly? Maybe. Or if I'm looking here, maybe there's other ways we can get to it, right? This silk, if it's stimulating polo-like kinase, what if I get rid of that? Maybe polo-like kinase won't be stimulated and there won't be a problem in the cancers. Or maybe this gene is basically inhibiting the destruction of this complex, which promotes angiogenesis, mitosis, right? What if I get rid of that? Now then we'll have a block to this. And the odds of me getting all of these gang members is 2 times 10 to the negative 8th, right? This doesn't happen by random chance. What this is telling me is polo-like kinase is absolutely playing a role in my system, and its gang is here, right? This is absolute more and more evidence. I'm about ready to go to court, is what I'm telling you, right? I got all this evidence. I'm excited. I'm like, all right, I got this evidence. We can get more, right? We also have specialized networks. So again, staying with our original, example, banks robbed. You've done the criminal history. Through that criminal history, you've looked for known criminal associations. What other information would help you? What do you think? Let me give you an example. All right. So say you have these individuals in the yellow, right? You want to see if they're guilty. Here's what you find is you don't find any criminal criminal associations with this group. But what you find is that this person works at, say, a auto repair shop. These three individuals work for them, and then this person's cousin, knows this person, right? So they all know each other. It's not a criminal association, but it's obvious that they all know each other, and they all said they didn't know each other when the bank was robbed, right? The odds that they might be guilty go up. And that's what happens a lot is, like I said, our ability to identify these pathways are only as good as our intel, right? But we do know that certain genes interact with other genes, right? It may not be, say a criminal act, but the fact that you have a bunch of genes near the bank that interact with each other would signify that, it's probably involved in the crime, right? It's these new gangs that show up. Mr. Pink, Mr. White, Mr. Brown. Do you guys know Reservoir Dogs? No one's seen Reservoir Dogs. This was back when Tarantino was actually good. Okay. Actually, I'm not going to recommend that one. It's bloody as heck. So, but again, it's the whole movie's about, they form these gang and they give themselves fake names to do this crime and it goes crazy. So, but that's exactly what happens. Like, we don't understand all of the relationships that happen. We see a relationship associated with a particular phenotype. That actually gets my attention. And we can do that. So what you can do is we're going to work on this. I'm going to demonstrate this program called String today. But what you can do is you can ask it, say, hey, find me the top 50 genes core or connected to polo-like kinase. And what it does is, it did that. And then you can see, well, now we've got this network that, isn't a specific pathway. It's just genes known to interact with it. What the heck do these things do? And if we look at some of these biological processes, so they're actually colored here, you can see most of these are cell division, right? G2 to M transition of mitotic cycle, cell cycle checkpoints, positive regulation of cell process, right? And a lot of these in the green are associated in the nucleus, right? Again, I found its associated gang, right? The gang that the cops know about, and now I'm finding this new association, which is probably the real gang, actually. and we can do that. And there's software out there. Has anybody heard of Ingenuity pathway analysis? No? Yeah. None of these labs around here have money, so no one pays for bioinformatics. But there's this program. What it'll do is if you put in a list of genes, what it'll do is actually generate these networks, and it'll actually introduce genes that might not be on your list, but actually can be used to connect the network. And so that's what I did was I threw a bunch of these pol-like kinase, the gang, through here. And what I find is that this VEGF comes up, right? And I don't measure it directly. But I see that it's connected to a lot of things that are actually changing in my system. Like the odds of all these associations are 1 × 10 to the negative 40th. Like this doesn't happen by random chance, right? So now as I've broaden out its network into things that the cops don't know now we've got another player here, VEGF, right? I've got another criminal, right? So here's pol-like kinase 1 and VEGF, and it's actually known previous interactions with each other. So that brings us to another example, and this will be my last one, I swear. It's master regulators. Right, so here's the example here. I've got 5 individuals here in the green. I looked at their criminal. They are close to the bank, which would maybe signify that they might be guilty. But I look at their criminal history. None of these have any criminal history. They're not in any gangs. They don't know each other. But they all take orders from this person here, this Godfather, right? Do you think the Godfather was anywhere near the bank when it was robbed? No, of course not. He's got an alibi. Yeah, I was at the movie. I don't know what you're talking about. That's terrible, Brando. I've been giving this talk for like years. You'd think my Brando would get better. So yeah, I mean, and this absolutely happens in, cancer research, is that something like VEGF, right? Or say your growth factors might be secreted somewhere else in your body, say your pituitary or, somewhere else, but it's in your bloodstream and it interacts with the genes that are, your tumor, right? You might not measure it directly, but with this stuff, we can actually see, like, we can find that Godfather just based on the evidence. Again, so here's what we do, and this is how we find them. And I think the easiest way to understand this is, say I'm by a pond, I throw a rock over my shoulder, and then I try to see where it lands, right? I might not see where the rock drops, but if I look over on the water, I can see the ripples it made, right? If I see the ripples, I can find where that stone that I threw is. We do the same thing in bioinformatics. So in this example, I have interleukin 1A is in the middle. It's a cytokine. It's expressed somewhere else, so you usually don't measure it directly. So I'm not measuring it directly, but here's what happens is I know interleukin 1A, it's been well studied. We know what genes it inhibits and basically stimulates. So in the red, any gene in the red means it went up in my system. In the green, it means it went down. These arrows, if it's an orange arrow, interleukin 1A is known to induce that gene. If it's a blue, then interleukin 1A is previously known to inhibit that particular gene. What I see in my list is I find 43 known targets of interleukin 1A, right? It's kind of like that, if I get 43 draws, what are the odds that I get like all these interleukin things? Pretty rare, right? So I know interleukin 1A is playing a role in my system, and I also know that given the fact that the vast majority is when interleukin 1A is known to activate it, it's going up, and when higher interleukin 1A is known to inhibit something, it's going down. The only 4 of these genes that are not obeying that are right here, right? Like most of everything that I'm looking at is screaming interleukin 1A is here. Not only that, but it's going up. It's more active, right? This is huge. It's like seeing the ghost. It's like throwing the, if there's a ghost around you, you throw up or invisible man or something, throw up some flour and it kind of lands on him and you see him. And not only do you see him, but you see where he's going. And that's exactly what we're doing with the bioinformatics here. I will also say that it's the really big players probably do not actually change transcription levels. Anybody here working on P53? No? Good, because it's hard. The thing is you normally don't see, you have to drop a bomb on like a cell, like a radioactive bomb, in order to see like a change in P53 activity, or actually its expression. It's kind of like that. If I want to get somewhere fast, am I going to build a car or am I going to call a cab? Right? I'm going to call a cab. And that's exactly what your body does, is that, you don't have time. These important things like P53 need to be active now. You can't build it from scratch. So what you do is you just keep it in the cell, and then say you maybe put an inhibitor on it, and then you release that when you need it, right? You phosphorylate it. You translocate that to the nucleus, right? You do something, but you don't build it from scratch. And so that's why this stuff is important, because it helps me get to those things, right? I might not see the change in P53, but I know what it interacts with, and I can see that like ring of interaction, and I can know exactly what P53 is doing. And that's just it's honestly, I know that looks complicated. It's very, very simple math. Like none of this stuff is that complicated mathematically. And not only can we do this for genes, we can do it for microRNAs, right? We know exactly what microRNAs, based on their sequence, what they're supposed to inhibit, right? And we can look for changes in those genes to determine what that microRNA is doing. If our microRNA goes up, I see all its targets going down, I could say it's probably going up. If all its targets suddenly increase, then we're probably losing that microRNA, right? And we could do this with chemicals. So, like it used to be, I'd analyze young and old people, and in the young signature, you would always see like a cannabis, like marijuana signature in the young people. Nowadays, not so much. Like both of them have it. In fact, sometimes the older people have it more than others. Everything you take, everything you imbibe, everything, it causes changes in gene expression, and we can measure that. And through that measuring it, like I don't measure marijuana directly or THC, but I can see the changes effects it has on the genes in us, and I can actually say that it's there, right? And we can also do this with drugs. So I give this example a lot. One of the most unusual clinical trials I was involved in, I don't know, especially like, does anybody working for an MD? No? Okay, oh good, I'll talk crap about them. Doctors are the worst at planning experiments, like the worst. Like they're good at treating patients, but ask them to plan an experiment and they just rush through it and they don't think things through. So they were gonna test this pulmonary hypertension drug. And so for this drug, for this clinical trial, they had a group of older men who couldn't be on anything except the pulmonary drug. Then what they did was just kind of find this group of just random control people, at least they were older men, about the same age, but they could be on anything else except the clinical trial drug, right? And then I basically analyzed those two groups and put it together. I did not find any traces of their drug, in the transcriptome, but what do you think I found? I actually found a drug in the control aged elderly men group. What do you think drug that I found in that group? What do you think? Elderly men.
Viagra?
Yes, vasodilators. It was huge. The signature was huge too, right? And so that was like the worst lab meeting I ever went to. Is like I had to go in there and tell them that their $1 million clinical trial drug was overshadowed by boner pills. They're like, really? I'm like, yeah, really. It's bad. Again, we see all of this. It's not only am I measuring things directly, but I'm also measuring the effect things have on other things. All right? And I'll go through this. So again, remember VEGF, here's our ringleader, found a godfather, angiogenesis, stimulates blood vessels, huge criminal history, lots and lots of cancers it's involved in. Again, you know, when I look at the genes, so genes correlated to histology, these are genes that actually get worse or increase or decrease as the cancer gets worse. In like 713 genes that I've found that are correlated to this, I found 48 targets of VEGF, and they're all given here. VEGF is known to induce pretty much all of these, and a vast majority are going up. So not only do I know VEGF is playing a role in my system, but it's also more active. So what I did was I took that average gene expression, I plotted all that stuff out. It kind of looks like a mess here. But then what you can do is you can kind of normalize all this, subtract the mean from each value, get everything on the same level. And now look how tight that is, right? I am willing to bet I could probably try to measure VEGF directly, but I bet based on all its targets, the average of all its targets, this is exactly what VEGF is doing in my cancer, my system. As the cancer gets worse, VEGF absolutely and utterly increases, and increases a lot as the cancer becomes cancer. Right? And we can also use this maybe to, again, can we use VEGF status or the targets of VEGF to predict whether we need to treat the cancer before it becomes cancer, or can we let it go? You know, and we can find its gangs. This is an example of one of those gangs. And again, you know, what kind of crime is VEGF committing? The worst kind, murder, right? So when you have high levels of VEGF, you're more likely to die. This is what we're trying to do. This is my goal, and it's, you know, is to find out all these like networks, these gene networks, because again, as I said, it is not just one gene, it's lots of genes, and it's how they work together that makes this important. And if you only focus on one gene, you're missing everything. I guarantee it. And I know there's lots of labs out there that do that. So don't be that lab. If you're focusing on a gene, you know, make sure you know its buddies, its enemies. And that's what we're going to do today, is I'm going to show you how to find those gangs without even having any data yourself. Like, there's so much information out there. Whatever you're working on, we can find information on. Right? Farther you enter the truth, the deeper it is. And that's the truth, right? I've never gotten into a situation where it just gets simpler and simpler. You know, it can always, you know, whatever rabbit hole you go to, it can expand, and it can get small and expand. And, you know, I honestly think, you know, it's just kind of like with a business. You can be a businessman, a salesperson, but the person that really understands what the business is doing is the CPA. You know, it's the accountants, it's the bookkeepers, you know, and I kind of consider this, you know, bioinformatics as kind of being that bookkeeper, you know. It's getting in there, getting into the numbers and figuring out actually what is going on, and then giving that information to somebody that'll actually do something with it. All right, well, let's take a break now, and